Hello everyone, my name is Prasanna Kumar Sahu, Assistant Professor, Department of Biochemistry, Dhabaleswar Institute of Pharmacy, Khuntuni, Katak, Odisha. This lecture will be on nucleic acids, which is present as a chapter in biochemistry syllabus in bpharm second semester. So what is nucleic acid? What is nucleic? A nucleic refers to nucleus originating from nucleus or associating with nucleus of a cell. Actually, when scientists first discovered DNA, they found it inside a nucleus. That's why they named it nucleic acid. So nucleic is anything that is originating from nucleus or associated with nucleus of a cell. Then what is acid? Have a look at briefly the schematic diagram of nucleus. It contains a nuclear pore. The nuclear pore is a small opening which controls what enters and exits the nucleus. Then the chromatin. Our DNA exists in the form of chromatin. Chromatin is as the name suggests, it actually a protein, a histone protein around which DNA is wrapped. So when DNA is wrapped around a histone protein, we call it chromatin. A question arises here, why don't our DNA exist alone? Why it exists with chromatin or a histone protein? The reason is our DNA is extremely long. One human cell has 2 meters of DNA, whereas the nucleus is around 5 to 6 micrometer in diameter. So since our DNA is extremely long, it cannot fit inside nucleus properly. So what the histone protein does here is it helps in packing the nucleus, packing the DNA so that it can be able to fit inside the nucleus properly. And another reason why histone protein is with DNA is it protects the DNA from radiation, from enzyme, or from chemicals. These two reasons, it protects the DNA, another one is helps in packing the DNA properly. These two reasons why histone proteins are with the DNA. So DNA is wrapped around histone proteins and we call it chromatin. What is nucleolus? As you can see, the nucleolus is a place where ribosomes are made. Who make ribosomes? Ribosomal RNA make ribosomes inside nucleolus. What is nucleoplasm? A nucleoplasm is a jelly-like fluid which surrounds nucleolus. What is nuclear envelope? The nuclear envelope, as you can see, is the outer covering of the entire nucleus which provides structural support to the nucleus on which nuclear pore is present. That's it. So this was the basic idea about nucleus. We understood what is nucleic, that is anything that is originated from the nucleus or associated with nucleus. If you look at the schematic diagram of nucleus that is currently on your screen, where is RNA present? We know that DNA is present as chromatin, but where is RNA present? RNA is actually produced by DNA through a process called transcription, and this RNA is present inside nucleolus as well as in nucleoplasm. Which RNA is present inside nucleolus? Ribosomal RNA is present inside nucleolus, whereas transfer RNA, tRNA and mRNA, messenger RNA, is present in the nucleoplasm. RNA is temporary because RNA is produced, DNA is only DNA its only job is to produce RNA to carry out protein synthesis. So whenever there is a protein need of the body, DNA produce RNA inside nucleus. After the RNA being made, the RNA exits the nucleus and enters cytoplasm to carry out protein synthesis inside ribosome. Got it? Next, what is acid? Nucleic acid. We all know what is nucleic acid is. Now what is acid? Please look at the structure of DNA. If you look at the DNA structure, there are three parts. One is that PO4 phosphate group, the red one, the phosphate group, the black one, the sugar moiety, and the green one or the nitrogenous base. But the acid is actually the phosphate group act as an acid. Why? Because phosphate PO4 is from phosphoric acid H3PO4, H plus PO4 3 minus. So since phosphoric acid can donate a proton, and according to Bronsted-Lowry theory, phosphoric acid is an acid. So since phosphate is from phosphoric acid, because H3PO4, H plus PO4 3 minus, PO4 3 minus is present here. So since phosphate is from phosphoric acid, this phosphate acts as an acid. That's why we call nucleic acids. Means originating from nucleus and the presence of a phosphate group, nucleic acid. So what is the definition of nucleic acid? Polymers. What are polymers? What do you mean by polymer? A polymer is actually, listen carefully, a polymer is actually a large molecule made by joining small repeating units. A polymer is a large molecule made by joining small repeating units. For example, train. A train has many coaches, isn't it? So these coaches are small units. They are all look same. They are repeating. So a train is a polymer. These coaches are small units which are repeating continuously. Another example would be starch is a polymer of sugar. Now many sugar moieties join together to form starch. Similarly, nucleic acid is a polymer of what? Polymer of nucleotide. Nucleotide refers to, nucleotide is actually consists of three parts. One is a phosphate acidic part, one is a sugar part, another one is a nitrogenous base part. So these three parts combinedly a single nucleotide unit. So polymers of nucleotide means, let us look at closely the DNA double helix structure. GC, TA, TA, GC, AT, CG, AT, all are repeating, isn't it? Similarly, the entire nucleotide unit is repeating, because nucleotide unit consists of adenine, thymine, cytosine, guanine, all are repeating. Since the nucleotide structure is continuously repeating, we call it a polymer of nucleotides joined together by phosphodiester bond. Phosphodiester bond. Look at closely the sugar moiety, the 5-member sugar molecule. One phosphate group, two phosphate group linked, PO4, double phosphate group linked. So phosphodiester, one phosphoester, two another phosphoester, phosphodiester. So from where ester came into picture? Ester refers to acid plus alcohol, it forms ester. So acid here is what? Phosphate group acid. Now alcohol is where? In the entire structure, where is alcohol present? The sugar. Why the sugar is alcohol? Because of the presence of multiple OH groups. The sugar act like an alcohol. So acid plus alcohol forms ester. So phosphodiester. And if you look the DNA double helix structure, there is a 3-5-. This is because at the third and fifth carbon, the phosphoester is linked. The third and fifth carbon, the phosphoester is linked. That's why it is written 3- and 5- linked by 3,5 phosphodiester bond. That's why we call. So what is the complete definition? Nucleic acids are polymers of nucleotides linked together by phosphodiester bond. That's it. In the next video, we will discuss its classification as well as its chemical nature and biological role. Till then, thank you. If you found this video is helpful, please like it, share it, subscribe it, and please do not forget to click the bell icon. Thank you.